Okay, we're going to discuss all about the gram-negative diplococci. So we have here the species of your Neisseria, and we have also your Moraxella. So generally, we have here the general characteristics of your gram-negative diplococci. So from the words of this is gram-negative and diplococci, so they are cocci. Most likely they are in pairs, diplococci. Okay, or rather they are more of the cocobacilli. Okay, so they are aerobic, so they have a strict requirement for the oxygen, and they die in the presence and absence of the oxygen. This one group also try to be oxidase positive, catalase positive, and they are non-motile, and they are capnophilic. So when speak about the capnophilic, they require 5 to 10% carbon dioxide at high humidity environment for them to survive. And they are also uh, saccharolytic, they tend to ferment the glucose. Aside from that one, some of them try to ferment also other sugar like your lactose. In the case of your, or even the maltose, except with the three species here, your Moraxella, catarrhalis, we're not able to be fermenting your carbohydrates. That one is non-fermenter. So we have here the pathogenic species of our Neisseria. So we have your Neisseria gonorrhoeae that's try to, you call it once your gonococcus. And we have also here Neisseria meningitidis, your meningococcus. And we have also your Moraxella catarrhalis. So we're going to discuss today all the three species here with their morphology, the disease association, and the different laboratory tests that will also help identify this different species of your Neisseria. We start the discussion with our Neisseria gonorrhoeae. So Neisseria gonorrhoeae is actually the causative agent of your gonorrhea, which is a sexually transmitted disease. So this is a venereal disease, meant to say that one is sexually transmitted disease. So before, this one is being called here as the flow of seed because the infection produced by this bacteria will try to produce here a highly purulent discharge. So it would resemble here the seminal fluid. That's why they call this one as your flow of seed. The French actually call this one as your clamp from the word clapoir or brothel. Brothel is something to be like, it's a beer house. So because again, this one is actually transmitted. Okay, so the infection caused here by your Neisseria gonorrhoeae is actually an acute pyogenic infection. So it's a pus-forming infection, primarily infecting here our genitals, including your endocervix, your urethra, and we have also here the anal canal. Basically, try to infect our non-ciliated columnar epithelial tissue found on our genital tract. Okay, we have here the different virulence factor associated with your Neisseria gonorrhoeae, which is responsible here for the pathogenesis produced by our bacteria. The first one, this bacteria would have receptor to the human transferrin. So transferrin is transport protein of your iron. With that, it tries to regulate and control the level of your iron in your biological specimen, biological fluids or the body fluids. And it might eventually try to deprive the host here with the necessary iron necessary for the growth of the host. Another one, this bacteria also contains here the outer membrane protein divided into three. We have the protein 1, you call this one as your porine. So it's a channel that will allow the entry of the nutrients of the host of the bacteria and also allows the exit or the excretion of the metabolic waste of the bacteria. This outer membrane protein responsible here for eventually protecting against the host and its complement activity. Complement help the host here in order for that to eliminate against the pathogen inside your body. So this outer membrane protein, the protein 1, prevents this bacteria not to be killed by the complement. Okay, so in the case of your Neisseria gonorrhoeae, it's made up of the pore B, porine B lang siya. But in the case of your Neisseria meningitidis, it's a combination of pore A, porine A, and porine B. The second one, we have the protein 2. This is your OPA, or simply opacity. So this is a protein that allows this bacteria to adhere or bind to your phagocytes in your epithelial cells of the host tissue in order for that to cause an infection. Okay, the protein 3 represents your reduction modified protein and try to block here the Fc portion of the IgG antibody, preventing this antibody to bind with your bacteria. Okay, then we have here other virulence factor related to your Neisseria gonorrhoeae. So we have here your pili. The pili would help here the bacteria in order for that to adhere or to bind to your host tissue. And they also try to use this pili here. We're talking about the sex pilus as their conjugation tube for their reproduction. So based on that, we can classify the strain of our Neisseria gonorrhoeae as to the presence of the pili or having the pili which are virulent. Okay, those are the T1 and T2 na strain. Your T3, T4, T5, on the other hand, do not have the pili and those are virulent. Another one, we have here your capsule. So capsule would try to protect the bacteria with recognition with your phagocytes, so therefore it could not be phagocytous. That would have an anti-phagocytic property. Then we have also here the LOS, lipo-oligosaccharide. Could also be found here on the lipid A moiety of the LPS of your gram negative because this one is still gram negative. So this is a type of an endotoxin. So when we speak about endotoxin, still part of the bacterial cell. So this one usually can be found on the bleb or the fragments that is being produced by the bacteria during its rapid growth. And within that fragment or the blebs contains this LOS, lipo-oligosaccharide, that's possible here for mediating the inflammatory response and at the same time allows bacteria also to cause damage to your host tissue. Okay, another one, we have your IgA protease. So they try to cleave or damage our IgE antibody. Okay, we have here the epidemiology. So the Neisseria gonorrhoeae is actually considered to be one of those leading causes of your sexually transmitted infection. The first one is your virus, na human papilloma virus, followed here by the bacteria Chlamydia trachomatis. And third siya, but in terms of the bacteria, so second siya, after your Chlamydia trachomatis. We have here the infection caused here by Neisseria gonorrhoeae. So again, it's trying to cause here sexually transmitted infection. For the male patient, that try to cause here symptomatic infection like your urethritis or the inflammation of the urethra or the infection in your urethra. And we have also here the dysuria with the painful urination with the presence of your pus. So may discharge ang patient natin. A symptomatic infection also try to occur here on the 3 to 5% of the male population. And those asymptomatic strain requires special growth requirement. So those are the Neisseria gonorrhoeae strain which require your arginine, hypoxanthine, and uracil as the growth requirement. Okay, for the female naman, it try to cause also here your symptomatic infection in the form of your, again, dysuria, cervical discharge, would have discharge coming from the cervix of the female infected na patient. And then it also try to cause here abdominal pain. For the asymptomatic infection, so that's related to your pelvic inflammatory disease. We call that one as your Fitzhugh-Curtis syndrome. And later on, the patient would have a complication of sterility, ectopic pregnancy, and we have also here peri-hepatitis. Another one, we have your conjunctivitis or the eye infection brought about here by the baby born out of the mother infected with this bacteria. During the delivery, this infection can be acquired by the baby, and that would result to your ophthalmia neonatorum. So that's because of the infected na birth canal ng mother. But in our healthcare settings, so all newborn, born of the mother, that are being given here by an eye drops antibiotic in the form of the erythromycin that can help here prevent the infection brought about by your Neisseria gonorrhoeae. Now we go to your Neisseria meningitidis. So let's try to cause here your meningitis. So we have here the different virulence factor. First, we have your pili as also for the attachment. And the most prominent na virulence factor related to your meningitis would be the polysaccharide capsule. Based on that, we can classify the strain of your meningitis as A strain responsible for the pandemic infection, BC for the community acquired, Y for the pneumonia, and W135 which try to cause invasive infection. This also contains outer membrane proteins just like your Neisseria gonorrhoeae. So made up of the protein, outer membrane protein 1, which is your porine. But this time in your Neisseria meningitis made up of the porine 4A and the 4B. Then also contains here the outer protein, the protein 2, which is your opacity, and the protein 3 in the form of your reduction modified protein. Another one also contains your LOS endotoxin. The same could be found in your Neisseria gonorrhoeae and IgA1 protease, which try to cleave hydrolyze our antibody na IgA found in our secretion. Okay, then we have here the clinical infection caused here by Neisseria meningitidis. So this one try to cause here meningitis, meningococcemia. So that can be transmitted primarily by respiratory droplets. Okay, so we have here the infection caused here by Neisseria meningitidis. So number one, it try to cause here endemic and epidemic meningitis. It's also try to cause here meningococcemia or sepsis. Primarily, the bacteria try to enter your body through your respiratory tract. Within your respiratory tract, that will be taken up by your nasopharyngeal macrophages where they try to multiply. And from there, they try to break with your blood vessels. So it try to cause here, once it try to already breaks up with your blood vessels, it try to cause here, can spread to different parts of your body, including your CNS, which try to cause here your meningococcemia. For our meningococcemia, so although some patient has been treated already, but there's still approximately 15% fatality pa din. So, ganun siya ka-deadly. Okay, manifestation of that, once it's trying to breaks up in the toxin here in the blood, so you call it once your meningococcemia. It also try to produce here petechial lesion or purpura, which is characterized by because of the hemorrhage in your blood vessels, which try to go to your skin and mucous membrane. And later on, it tried to develop here a tachycardia, and we have also thrombosis or the clot formation that might develop here to your DIC or disseminated intravascular coagulopathy. Another complication of this one would be your adrenal gland hemorrhage. We call that one your Waterhouse-Friedrichsen syndrome. Now we go to your Moraxella catarrhalis. Okay, so this virulence factor includes here the adherence that would allow this one to adhere to your upper respiratory tract part of your body. So we have here the epidemiology. So Moraxella catarrhalis here commensal. So this one could be found normal in a healthy patient in the upper respiratory tract in the primarily young children, adult patients, but in the lower respiratory tract, it try to cause here chronic obstructive pulmonary disease. Okay, then we have here the clinical infections. So this is third leading cause of your sinusitis and otitis media among children. So it also tries to cause you systemic infections in the form of your endocarditis, in the form of your meningitis and tracheitis. Okay, we have here laboratory diagnosis of specimen in order for us then to diagnose infections. Neisseria gonorrhoeae, since this one is sexually transmitted infection, so our genitals by swabbing. Okay, that's the specimen for that. So you can swab the urethra by inserting the swab 2 centimeters, or you could have here your anal canal 5 centimeter. You can also have the endocervix for the female patient. Try to swab that one. You could have we have your rectum and pharynx as our specimen. Neisseria meningitidis as one try to cause meningitis, so our specimen for diagnosis, you could have your CSF. You can perform also the blood culture or nasopharyngeal swab. Moraxella catarrhalis on the other hand, so this one would have here the middle ear effusion, nasopharyngeal swab, sinus tract aspirate, but also sputum aspirate or bronchial aspirate are as your specimen to diagnose the infection. Okay, we have here the specimen collection. So when we are collecting the specimen, so basically the best here is to plate that one directly to the selective medium. But if not, then you can have your transport medium, provided here that when you try to receive that specimen in the laboratory, then you need to plate that one directly to the selective culture media. So for the specimen here, because we are collecting that one through your swab, okay, so okay, we can use here your rayon or dacron swab instead of the cotton tip or your calcium alginate na swab because those are toxic to your bacteria. And most likely, this bacteria also try to be highly sensitive here to the drying. So once your specimen dried up, it would eventually it would kill already your microorganisms. So be careful with that. So you can have your selective media in the form of your thiomartin, which is your chocolate agar base. Then you could also have your transport medium in the form of your Gembic or the James A. Martin biological environmental culture medium. That would It's a commercial system that would transport system that would allows that one to provide here the necessary 5 to 10% carbon dioxide requirement. Okay, we have here the microscopic identification. So this would appear to be gram-negative diplococci and would appears to be, in the case of your Neisseria gonorrhoeae, it appears to be in that one is in pairs, forming here your bean shape or coffee bean shape. We call it once your gram-negative intracellular diplococci because this one could be found within your WBC. And that's considered to be a true infection. But sometimes it could be present outside of your cell. You call it once your extracellular, especially during the early infection. Okay, we have here the culture media to provide the necessary 5 to 10% carbon dioxide here. So you could have your candle extinction jar, or you could also have your carbon dioxide incubator, providing the 3 to 5% carbon dioxide. I sorry, that's not 5 to 10%, but rather 3 to 5% carbon dioxide. Okay, with a high humidity environment. Okay, we have here the different selective media. So these are your chocolate base. So the first one, we have your thiomartin, so primarily incorporated with the following antibiotics. So VCN, vancomycin, which is inhibitory to your gram-positive colistin. This is inhibitory to your gram-negative. And we have also the N-nystatin, which try to suppress the growth of your yeast. Another one, you could also have your modified thiomartin where it's being incorporated with your trimethoprim that will further inhibit here the growth of your proteus. Then we have also here your Martin Lewis. That one would have the VCE or made up of the vancomycin, colistin, and anisomycin as a replacement for the nystatin in your thyromartin. Then we have also here your New York City agar. So it's made up of your vancomycin, colistin, and A for the amphotericin. Okay, as a replacement for the nystatin in your thyromartin and anisomycin in your Martin Lewis. Plus, meron pa rin siyang trimethoprim. Then we have also your G-Select agar. So made up of vancomycin, colistin, amphotericin B, plus trimethoprim plus lincomycin as an additional inhibitory substance, antibiotic here for inhibiting the growth of your gram-positive na bacteria. So incubation, so this is 35 to 37 degrees Celsius here, the growth temperature requirement, maximum temperature requirement for the Neisseria, providing here the 5 to 10% carbon dioxide. Sorry, that's your 3 to 5%. Okay, that's a 3 to 5% carbon dioxide up to 72 hours. On the other hand, your Moraxella here able to grow at a maximum temperature, lower temperature. That's 28 degrees Celsius at 3 to 5 degrees Celsius, 3 to 5% carbon dioxide also at a high humid environment. Okay, then we have here the colony characteristics. So we have Neisseria gonorrhoeae appears to be small, tan, translucent, and maybe raised. Neisseria meningitidis would also be small, tan, but that one is more of convex and highly mucoid because of its capsule. Moraxella catarrhalis would appear to be smooth, opaque. That one would be gray to white and can be, if you try to push it one with your loop, okay, the colony here would remain intact. You call it once your hockey puck appearance. And after 48 hours would have your wagon wheel appearance or a wagon wheel colony characteristic. Okay, then we have here the presumptive identification, the biochemical reaction. The first one, we have the test for the oxidase. Again, all your organism here, your Neisseria and Moraxella are all oxidase positive. So we have here your indicator 1% dimethyl paraphenylenediamine dihydrochloride. Another test, we have the superoxyl test. This is your catarrhalis test, but this one uses here 30% na hydrogen peroxide. Superoxyl test will only give a positive result with your Neisseria gonorrhoeae. Then we have your carbohydrate utilization with the fermentation of your sugars. So you can have your CTA cysteine, trypticase agar that would allows you to primarily determine if the organism here try to ferment your glucose, your maltose, or lactose. So we have your phenol red as your indicator for that. Yellow is a positive color, red is a negative result. So Neisseria gonorrhoeae will be fermenting the glucose. Neisseria meningitidis will try to ferment the glucose and your maltose at the same time. Whereas your Moraxella catarrhalis, we consider that one as your non-fermenter, so therefore it will give all the negative result. Another test, we have your quad firm test. So again, it's made up of your four na sugar, glucose, lactose, maltose, and sucrose. Also, it will also allows you to identify the denase. So, meron siyang denase na enzyme or you could also have here your beta-lactamase. Okay, we have here the summary of all the three species here. So, the first one, superoxyl, 30% hydrogen peroxide will only be positive with your Neisseria gonorrhoeae. Your selective and culture media, your thyromartine, modified thyromartine, so all of them were able to grow on that. Again, this is a chocolate agar base. Blood agar will it be inhibitory to your Neisseria gonorrhoeae. It will not allow the growth of your Neisseria gonorrhoeae, but it will have here growth of your meningitidis and Moraxella. So we have the fermentation. So again, Neisseria gonorrhoeae only ferment the glucose. Meningitidis try to ferment the glucose and maltose. You can have your others naman. Lactamica try to ferment the glucose, maltose, and the lactose. Zika, mucosa try to ferment the glucose, maltose, and we have also here your sucrose. Then we have here your denase test, and we have also here your butyrate esterase test with the substrate your tributyrene. So this one will only give a positive result with your Moraxella catarrhalis.